Hiya and welcome to Gene Dr. Qais Abu Ali. In today's episode, we'll be discussing some of the principles, concepts, and genetics. I'll be talking about different types of screening or testing, different types of samples, ages. We're talking about some different types of mutations or variants, also some different types of modes of inheritance. Many of you have heard of DNA, gene, chromosome. I try to simplify how these three are connected with each other. Let me start with the DNA. DNA stands for deoxyribonucleic acid, DNA, and essentially it's the genetic material that we as humans, as individuals have. This is the double helix structure, and each helix has a sugar, deoxyribose, as well as a phosphate, as well as a nitrogen base. And this nitrogen base is a letter that is one of four. A, or adenine, C, or cytosine, G, or guanine, T, or thymine. That is called a nucleotide. And those stretches of nucleotides form the DNA. And that's found in each helix. So for the double helix, each of these base pairs or nucleotides attaches with its counterpart. So for example, A or adenine attaches or connects with T with thymine, and cytosine connects with guanine, C with G. And as humans, we have about 3 billion bases, and 99% of these are identical. So those stretches of nucleotides form genes, or we have about 20,000 to 25,000 various genes. Those genes would encode generally to various types of proteins. Some of these genes have regulatory function. And as these stretches of DNA inside the nucleus of the cells get packaged and attached to a protein called histone, this packaging forms in the end the chromosome. Nearly 100 years ago, we used to think that as humans we had 48 chromosomes. And then that understanding has improved so that we found that as humans we have 46 chromosomes, or 23 pairs. And out of these 23 pairs, we have 22 pairs that are called autosomes. And the last pair, or the 23rd pair, is an XX or an XY or sex chromosomes. And that's what differentiates men from women, males from females. Each one of these pairs is inherited from a parent, and each chromosome has a copy of a specific gene that is inherited from those parents. Granted that we're talking about genes that are on the autosomal chromosomes, or the 22 pairs. So when we're talking about various types of genetic diseases, we're talking about in general, either a chromosome abnormality or a gene abnormality, just to simplify it really. When we're talking about chromosome abnormality, that could be abnormality in the numbers, 47, 48, 45, etc. But also, we can see genetic diseases when there is an increase or decrease or an alteration, a part of that chromosome. So for us in genetics to understand the number of chromosomes in an individual, we have to have a procedure or a test to enable us to look at that number and the X or Y presence in those individuals. And when we look at those chromosomes, that is what is called a karyotype. 46, the number, and XX for a woman, and XY for a man. Anything other than that usually does result in genetic conditions. When we look at diseases caused by genes, we have genes that are really small, could be 100 bases, to genes that are millions of bases. So for us to look at these genes, we have to understand the letters in that gene. Think of our genetic material as a library, and that library has different rooms, different chromosomes. Each room has different shelves, different genes, and those genes have these books that have those letters and words, or the sequence of that DNA. So as you can imagine, for a book to make sense, those letters have to be in a certain order. Sometimes that typo might result in a completely different word, or a completely meaningless word. Some other times that typo might not necessarily result in something significant. And that same concept applies really to changes in these letters. And that is what is called a mutation or a variant or an alteration, which is a change of the normal sequence that we see. 99% of our DNA is identical as humans, and that 1% is what makes difference. As humans, we do have these typos, if you will, that would not necessarily result in genetic diseases. And these typos, what we call SMP or SNPs, when there is a specific disease, sometimes there is a specific gene that gives us those features of that disease, or there is a polygenic disease or a disease that is resultant from multiple genes. And there is a huge number of diseases that do fall under a single gene or polygenic, multigenic diseases. Part of the genetics practice, we tend to see patients in all age groups basically. Females during their pregnancies, but also the fetus. So that is the prenatal period, and also the postnatal period, which means when the fetus is born, then as they grow up as kids, and then as adults. Also, there's a step before that we could obtain genetic testing and results for certain mutations in certain genes. And that's even when the egg gets fertilized, and that's what's called pre-implantation genetic diagnosis, or PGD. When we talk about the types of samples that we would like to test, we could run genetic tests on a blood sample, various tissues, but also we could run it prenatally or during the pregnancy, inclusive of amniotic fluid, or the fluid that surrounds the baby or the fetus at the time, and also what's called the CVS, chorionic villus sampling, or part of the placenta during pregnancy. Sometimes there's what's called a screening test. Usually is a quick test that provides a certain amount of information that helps identify the amount of risk that a disease could occur. There's a test that is called NIPS and IPS, non-invasive prenatal screening, and it looks at various risks for various chromosome abnormalities. If the risk from these screening tests is high, then the next step is a diagnostic test, and the diagnostic test during pregnancy is looking at samples, for instance, from amniotic fluid or the CVS, and we run the specific test that would address the findings from the non-invasive prenatal screening or NIPS. But also during pregnancy, if there is an abnormality, when there is a known genetic disease in a specific family, and we end up knowing what that chromosome change or that gene change or mutation is, we could test that same gene change and alteration or mutation in a fetal sample, and that's from, again, the amniotic fluid or from part of that placenta, the CVS. When I see a patient who has various features, and I think of a specific genetic disease that is caused by a specific gene, then it is easier to get testing for that gene. Or if it's a group of genes that cause that disease, or a few diseases that do have an overlap in clinical features, then we would end up in sending what's called a panel test, for example, where there are multiple genes. When we find an answer, whether that's being caused by a gene abnormality or mutation or a chromosome abnormality, then the next step is to provide the answer to those members and their families, and try to find a solution, a treatment, a clinical trial to be involved in if there's no treatment, and try to help them with various assessments, various modalities if they need them, hearing evaluation, heart evaluation, eye evaluation, etc., etc. And that's when we start talking about what's called a mode of inheritance. In autosomal dominant disease, all it takes is just one copy to be affected or have a mutation for a disease to present itself. You don't have to have two copies being impacted. So you could have one normal copy of the gene on one chromosome and one mutated or abnormal copy of that same gene on the other chromosome. In recessive diseases, one has to have that gene mutated or altered on both copies of the chromosome, meaning that both genes on both chromosomes have to be affected or impacted or having a mutation. It doesn't necessarily have to be the same mutation or the same change in those letters, as long as there are two changes. If there's an individual who has a variant or mutation in one copy of the gene but not the other, and he or she doesn't have a disease. Most of the time, that is what is called a carrier state in a recessive disease. So they carry that mutation, but they do not exhibit those symptoms. They don't exhibit that disease. They're not affected by that disease. That individual could potentially in the future pass on that mutated gene onto their offspring. I'll be posting the same episode in Arabic language. If you're interested in learning more, make sure that you subscribe. If you have any comments, please post them down. And until we meet again, thank you, stay well, and be safe.